Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Welcome back to Neuroverse, and welcome to another episode of our Tools for Looking into the Brain series, which is sponsored by Scientifica. Today, we are joined by Dr. Sasha Alez, an associate professor at the Department of Anesthesia at the Cincinnati Children's Hospital in Ohio. Sasha first obtained an undergraduate degree in biochemistry at Imperial College London, followed by a PhD in neuroscience at the University of Alberta with Professor Peter Smith, where he began researching the mechanisms underlying pain, specifically focusing on the electrophysiological mechanisms of gabapentinoid drugs in the spinal cord for neuropathic pain. He then became a postdoctoral fellow in two places, at the University of British Columbia with Professor Terry Snutch and at UCL with Professor John Wood, where he studied ion channels involved in nociception and chronic pain. And in 2019, Sasha joined the University of New Mexico Health Sciences Center before moving his lab and joining the Cincinnati Children's Hospital as an associate professor earlier this year. His group's current research focuses on understanding the mechanisms of chronic pain and developing new therapies for treatment. And today we'll be finding out a lot more about the current projects in his lab and about the tools that we can use to understand the elusive but extremely important topic of pain. Welcome to the podcast, Sasha.
Thank you. Thanks for having me.
I'm really excited that we are having an expert on the neurobiology of pain here today because I think this is actually a topic that we haven't covered much in Neuroverse so far, but also I personally was made aware pretty early on in my own studies how little we know about pain and how it is one of the most complex forms of perception that exists, both biologically and psychologically speaking. So could you tell us a little bit about how you first got interested in neuroscience and your specific research topic?
Yeah, so as you mentioned, I started off my career as a biochemist, just interested in basic science. And I did a very interesting internship at Imperial in the Division of Cell Biology, where I mainly looked at mechanisms of neurotransmitter release and exocytosis in different cell types. And so I became very interested in looking at that, and I became exposed to different techniques for studying neurons like calcium imaging and confocal microscopy. And so I just became fascinated by looking at release mechanisms. And so when I was delving into that sort of research, I happened upon a PhD opportunity back in Canada, where I'm originally from, to study mechanisms of neuropathic pain that was utilizing similar techniques to understand mechanisms of release of neurotransmitter in the spinal cord. And as somebody who sustained an injury playing cricket when I was in high school, I have had chronic pain for some time, and so I was acutely aware of the clinical relevance and importance of chronic pain. And so I kind of married the two areas of interest, and it just made sense to follow that in my PhD research and to continue to study mechanisms of release of neurotransmitter from primary afferent neurons, in that case, DRG neurons in the spinal cord. And I spent a lot of time looking at the spinal cord and studying spontaneous activity in the spinal cord in neuropathic pain models.
Yeah, really nice. I find this is like quite a neuroscience specific thing. Maybe when I hear people's like origin stories, it tends to be like I had this random scientific interest and then I linked it to my personal life and experience on like a more behavioral or clinical level to come up with this research topic. And I like how you melded your interest in like neurotransmitter release, which is something so specific, but also to make it applicable to something clinical. So, could you give us a brief overview of like what the main mechanisms of pain are? I know there are probably a multitude of different mechanisms, but yeah.
Yeah, so what we basically study are mechanisms of chronic pain, more recently acute pain we've been looking at. So this is basically a type of pain, specifically neuropathic pain, that persists for a very long time in patients. It depends on who you ask, but it's basically pain that lasts longer than it should, several months long, and it's intractable in many cases. It's not responsive to treatments like opioid drugs, which makes the issue of treatment of chronic pain kind of two-pronged, because you have the issue of intractable pain. People can no longer do their jobs. It's also sometimes called high impact chronic pain. So, roughly 1 in 5 people suffer from chronic pain, and about half that number suffer from so-called high impact pain, where they can't do their day-to-day activities. So you have all that lost productivity. People can no longer function in society. But then you also have the issue of opioids, which are highly addictive and have high abuse liability, and they're not very effective. So people become addicted to these drugs that are not effective, and it has resulted in the so-called opioid abuse epidemic. And so as a result of that, we have two issues in the field. We have the issue of chronic pain and the issue of opioid abuse resulting from it. So my lab mainly focuses on the study of non-opioid therapeutics for chronic pain. And so pain is generally thought to arise. So in the case of an injury, for example, you have an increased excitability of these peripheral nociceptors, primary afferents, which drives increased excitability along the whole circuitry, essentially, the nociceptive axis that results in increased excitability. And nociception is not to be confused with pain, because pain is an emotional sensory experience and it's very specific to the patient. But I study types of chronic pain that are essentially driven by peripheral activity of these nociceptors caused by things like traumatic nerve injury or damage to neurons in the case of different neuropathies like chemotherapy-induced peripheral neuropathy, patients who are treated for cancers. These chemotherapeutic agents result in damage to the sensory neurons, which again causes another issue of peripheral neuropathy that can be incredibly painful for people. And we study different aspects of chronic pain. So because we study the neurons and the interaction of neurons with the different cell types, we're studying different modalities. We're trying to understand different aspects of the pain. So some patients you speak to may know someone with chronic pain or have chronic pain yourself. Will have heard people describing pain as having a stabbing sensation or a shock-like sensation or a burning sensation. And so these different sensations are attributed to the activity of different neurons. And so that's what we try to understand in our models, is how we can effectively attenuate some of these painful sensations and better develop treatments for patients. And so, yeah, that kind of leads into our other sort of major research focus, which is the use of human tissues in pain research, which is becoming more and more important now because rodents are incredibly important for understanding behavior, but they don't necessarily predict how humans and human neurons are going to behave in a certain pain condition in a patient. And so it's becoming more and more important for us to obtain human tissues or human derived tissues to tease out mechanisms of pain and chronic pain.
I really like the distinction you made between the subjective experience of pain and then the physiological attributes to it. Could you explain a little bit more what is neuropathic pain and nociception? So are those, like you mentioned, electrical signals that you can record to identify whether or not someone is in pain? Because am I correct in saying that we don't yet have I guess, a measurement for pain, so you can only really go off of the subjective experience of the patient?
Yeah, that's accurate. It's been sort of the big sort of holy grail in our field, if you will, is to have an objective measurement of pain. Other than, it hurts when I do this, or Rachel's pain on a scale of 1 to 10. So we don't have an objective way of doing that. I know a lot of people in the functional imaging field who put patients in MRI machines are trying to come up with better ideas, signatures in the brain of how to understand that. That's completely outside my wheelhouse. But just from trying to understand chronic pain in general, this is sort of where we're at. And it becomes even more complicated when you try to model everything you're doing in a dish, which is where I sit. So, I mean, if you talk to a clinician, you'd basically say, I only study nociception. That's as far as I can go. However, we do have animal models of chronic pain, and we generally will allow the pain to persist for several months. And this is, of course, all in accordance with our institutional animal approval guidelines. so that we can better understand truly chronic pain rather than something that is sort of inflammatory pain or an acute pain like a pinprick. And these are two distinctly different processes that are involved in all of these stages of the emotional experience of pain. So nociception is not pain, but neuropathic pain, for example, is something that's caused by damage to the nervous system. It can be peripheral or centrally mediated. There are many different factors involved, and there are many different flavors of chronic pain. So it's a little difficult to answer that question in a succinct manner, but just suffice it to say that it's complex and involves many different mechanisms. Both neurons and other cell types are involved, including Schwann cells, fibroblasts, and immune cells that infiltrate the DRG. So studying neuroimmune interactions is becoming extremely important to better understand chronic pain. And then we have sort of the other elephant in the room, which is sex differences in chronic pain. Men and women are different. We're seeing that even at the level of rodents in terms of the immune mechanisms involved, roles of microglia versus T cells. These all have differential roles in male and females. And so developing treatments that are sex specific may be more important.
Right, and I guess that highlights the importance of what you mentioned, doing human work. So in a dish, even though that might be really hard to link directly to the subjective experience of pain, I guess you could look at sex differences more effectively there because you could compare, for example, like induced pluripotent stem cells from male or female donors, etc. I noticed a lot of your previous research, at least, was focused on the electrophysiological properties of dorsal root ganglion neurons or sensory neurons. And I was just wondering where exactly this fits in with the process of pain, because is it seen as a sort of downstream outcome where a lot of mechanisms converge to create differences in excitability that then causes pain? And so is it therefore like useful to compare pain by using a biological measure in these stem cell models? Or is it one of the first things that happens that contributes to downstream further mechanisms that lead to pain?
Yeah, so that's a chicken and egg question, right? So essentially, so both are correct is the answer to that. So we see increase in excitability early on, and then in certain pain states, we see the maintenance of that hyperexcitability driving pain states. However, it depends on what type of pain. For example, in the case of something like a spinal cord injury, we see something where the spinal cord is injured and it basically sensitizes the spinal cord directly, and then you can have sensitization of the primary afferents, the nociceptors below the level of that injury. So basically coming from the central nervous system back out again and driving neuropathic pain states. Whereas in the case of a peripheral nerve injury, so something to the sciatic nerve, for example, you see increase in excitability in the periphery early on, and that continues, but then that will bleed into the central nervous system, causing increase in excitability in the spinal cord and in the brain. So eventually everything becomes sensitized. Of course, it becomes important to consider sort of the gas pedal and the brake, if you will, in these circuits. So centrally we have descending modulation, and everything gets a lot more complex. For the purposes of my research questions, I mainly look at the peripheral mechanisms, things in the spinal cord and things that are released into the spinal cord. And I more or less stop there because there's still a lot to understand about what's going on at the level of the DRG. There's also a lot of real estate, if you will, from your fingertips all the way to your spinal cord. So these are incredibly long fibers. So there's a lot that can happen locally at the site of injury and along these axons, and then all the way to the DRG and the T junction before you ever even get to the spinal cord. So there are lots of things to understand about what's going on in these different anatomical regions in different pain states.
Right. And when you model that using stem cells, can you model it beyond like single neurons? Can you actually create a circuit? And with the neurons being so large, like I guess physically it's hard to recapitulate the true nature of these neurons.
So we haven't done that, but I am aware of other groups that are working on sort of microfluidic devices to sort of model that, where you have the primary afferents on one side and the dorsal horn neurons on the other, and you sort of try to recapitulate the first synapse in that pain circuit and sort of stimulate one side and record from the other and see what's going on in a stem cell model. So that is possible, and then you can do fancier things like supplement with different cell types, and see what the roles of specific cell types modulating the DRG neurons are. Lots of different things have been done in the past in that regard.
And I was also reading about your comparative study of comparing human-induced pluripotent stem cell derived sensory neurons and then the human dorsal root ganglion neurons. Could you tell us a bit about the differences between these two types of neurons to model pain?
Yeah, so there's a number of different factors. So HIPSC derived sensory neurons are easier to obtain, easier to work with, and sometimes easier to standardize. Although many groups will do things slightly differently from others, and so you'll result in your own iPSC derived system.
Sorry, those are peripheral sensory neurons,
That's
right?
right. Yeah. They're pretty good in terms of looking at overall excitability and firing with some subtle differences that we found in terms of action potential waveform properties. But human DRG neurons are from recently deceased donors. There's the variable of course, time after death in these donors and cause of death, which can complicate things. There's more donor to donor variability with the use of these primary human DRG neurons, but it is the primary system. And so you see differences in the exponential waveforms and the roles of other cell types that you perhaps would fail to see in the stem cell model situation. I will say, though, in the time since I began my training, I've seen remarkable strides in the development of better stem cell derived sensory neuron models for the study of pain. And so, the future is getting brighter in that regard. I think it will require a lot more effort and time from investigators to better compare against primary DRG neurons and perhaps recapitulate some of the sex differences that have been observed with primary DRG neurons and the role of other cell types along the neuroimmune axis, things like that. So.
Going back to the differences between acute and chronic pain, do you see mechanistic differences at the cellular level? Like, are the same neurons employed? Or is it just the electrophysiology that is different? Like, how does acute pain become chronic?
So I don't really look at the transition per se from acute to chronic. There are many labs that do that, but earlier on, acute pain normally attributed to the A delta fibers and the chronic pain attributed to the unmyelinated C fibers. And so these have different response profiles. This is a very old work from some electrophysiology groups that have characterized the roles of these different neuronal cell types. I will say one of the sort of topical things. I know I've said it several times now, but the role of different cell types has been largely ignored in these different transition pain states. So I think what has become clear is that in that transition, we have a role of different mediators released from other cell types that are driving the changes in the neurons that sort of switches the neuron from acute to chronic pain and then maintains that chronic pain state. And so earlier on, it's more a direct injury that's causing it to become excitable. We have tried to model sort of acute. You can do things acutely in a bath, applying it directly to a neuron, or you can leave it in culture for several days to try to model a more chronic situation to better understand what's going on. Different cytokines, interleukins, for example, can change the response properties of these neurons over time, and these will differ acutely versus chronically. So it's not a question of whether it's the same neurons. They may all be involved because you have those neurons that are innervating the same areas, but it's kind of the context of the injury and the exposure time, what happens to these different mediators that are driving the different pain states. So we have to think about time as a variable because that has largely been overlooked, and still you see many papers trying to sell chronic pain as something that happens 7 days after an injury, which is obviously not chronic, not at least in terms of what humans are experiencing.
Yeah, that's very interesting. In our podcast episode, we always come back to structure function relationships. And I think another way of thinking about it is looking at time. And I've read a bit about how patients that have chronic pain, it actually changes the structure of their brain. So, like different brain regions become more hyperactive or less active. So, I think it's interesting that you can see these structural and functional differences across these scales, not only at the single neuron level, but also wider in terms of the activity across different brain regions.
Yeah, and I think it is also interesting that pain per se can be actually quite useful. Like if we had no sensation of pain, then we would probably die because we wouldn't be able to tell when we're in danger. But I guess chronic pain is essentially just a maladaptive form when it stops being useful.
Exactly. It's a sort of form of pathological learning. That's sort of the best description I've heard before, is that basically these neurons learn to become hyperactive and hyperexcitable and sort of drive that pain state. And there have been studies to sort of make the neurons unlearn. And so the things that make the neurons learn, if you target those processes, for example, protein synthesis or things like that, you can actually prevent these changes in hyperexcitability. It's also interesting to think about that initial question you asked, the chicken or egg situation. Some people are playing with more fancy techniques like optogenetics to chronically stimulate the neurons, and they can actually drive a chronic pain state by just activating the neurons without actually injuring them per se in a traditional sense. So it has been shown that it is sufficient for neuronal activity alone to drive sort of a chronic pain-like state in rodent models, at least.
This may be slightly tangential to your specific research, but I was wondering if you know whether there are certain neurons or certain people that have a higher predisposition to having chronic pain. Like, is there, I don't know, like a genetic predisposition, or is there some type of like baseline firing rate that these neurons then, like you say, learn in a neuropathological way where they've just become chronic pain?
That's a difficult question because I'm not a psychologist, but at least from readings in the field, there are so-called catastrophizers and non-catastrophizers, people that will report higher pain scores, but when you look at their injury on scans, do not appear to have much wrong with them. Whereas somebody who comes in who's been used to dealing with chronic pain for some time, for example, someone who has served in the military, they may be reporting lower pain scores, but have like multiple disc fractures, for example. And so we have that emotional experience factoring into it. It's personal. I will also state that the IASP, the International Association for the Study of Pain, has changed their definition slightly of pain, in that you don't need to have anything on a scan in order for patients to actually have chronic pain. So in the cases of fibromyalgia, for example, it's very important to take that into account. So it can be pain resembling that associated with chronic pain that can actually result in that experience of chronic pain. So one of the research questions that brought me back to the UK after I did my PhD and my first postdoc to work with John Wood was to understand the mechanisms of NAV 1.7, a sodium channel, in which loss of function is associated with people having the so-called congenital insensitivity to pain. And so there are mutations in people that will predispose them to either feeling no pain or feeling no certain modalities associated with pain. And so his group, also in collaboration with some clinical geneticists in Cambridge, have been studying for many years different mutations associated with people that experience differences in nociceptive processing, either lack of pain or extreme pain disorders. And so it sounds a little bit like X-Men, but it's like mutants living among us sort of thing. Like these studies identifying mutations is an extremely costly thing to do, right? So you have to do whole exome sequencing. It has to be bad enough that you're going to see a clinician, and a clinician has to have the wherewithal to actually go and further study it and to collaborate with clinical geneticists. So I think there are lots of people out there who perhaps have some sort of mutations and sort of loss of sensitivity to pain or experience less pain. And that may also explain differences in response to different analgesics. We've shown that a standard of care therapeutic for treating neuropathic pain, gabapentin, actually shows different effects on human donor neurons. And that's with a relatively small group, but it's interesting nonetheless, and it actually is consistent with what clinicians have observed with gabapentin is that, different patients will experience different things. And so it's a personalized thing. So those might have a genetic basis. And we've known for a while also I work in an anesthesia department, so I talk to a lot of anesthesiologists. And so there are certain people that, for whatever reason, some anesthetics don't work as well. And so they have to sort of look at those patients more carefully and identify different variants, gene variants in those patients and see why it is that they don't respond to them or why they may be especially sensitive to them. So a personalized approach, as with most diseases, is probably very important.
I also wanted to ask, because it's been mentioned a few times now, about the variability of all the different cell types that are involved in this process. And I read one of your papers that was on the neuroimmune mechanisms in a chronic back pain model that was modeled in mice, and noticed that there were both electrophysiological changes found in the dorsal ganglion neurons, but also changes in the production of immune cells. And I just wanted to ask, do you think that these two processes interact mechanistically and how? Again, I'm still tapping into that chicken and egg question. But yeah, I was just wondering how immune cell activity or any chemicals that are released by these immune cells or any other downstream responses could interact with changes in neuronal excitability.
Yeah, so that's kind of an open grant question that I'm not gonna go into too much detail, but I will just say that there obviously is a role for peripheral macrophages in male mice, and so that is likely contributing directly to the effects on neuronal excitability. We have also analyzed some of the cytokines that are associated with macrophages from these mice and have found a few interesting players that might be involved. Some of them classic little cytokines that have been already well characterized, and some of them not. We've also looked at RNA sequencing data from these mice and have found a few interesting candidates that may be involved in regulating that sort of neuroimmune interaction. That work is actually a preprint from my former PhD student. And we're building on that currently to try and better understand neuroimmune mechanisms in this model of chronic back pain.
So, do you think that the sex differences that you've mentioned a few times now, in chronic pain could be explained by differences in the immune cells between sexes?
Yeah, at least partly. There's been a lot of noise in the field for the past few decades addressing that, and it seems to be largely attributed to immune cells. However, there's some recent evidence suggesting that neurons themselves are differentially responsive in males and females based on things like prolactin, for example, show different response profiles that may be attributed to certain female-specific chronic pain conditions like endometriosis, for example. But in the literature in the past, there have been studies that have examined the roles of microglia versus T cells. For example, we did a lot of work studying the role of P2X4 receptor. Mainly, we were validating an antibody therapeutic. And we found with our collaborators that these antinociceptive effects were only in male mice, which is consistent with the work of others, including Mike Salter and Yves DeConnick's group, and that female mice are not responsive to P2X4 receptor targeting therapeutics. And this is largely because of differences in the roles of microglia versus T cells and males versus females. And even in our own studies of chronic back pain, we found that the role of peripheral macrophages were prevalent in male mice with chronic back pain and not in female mice. And so we think that that's at least partially contributing to the differences in the mechanism. At least what we think is the males and females will develop chronic pain, so they'll develop similar levels of pain in the models that we look at, but they'll have different roads they'll use to get there, so to speak. Those roads being the different cell types and immune cells.
Yeah, that's interesting. And I was also thinking, you mentioned chronic pain can be in the central nervous system or the peripheral nervous system, right? And I know your work and what you've been talking about is focused on the peripheral nervous system, but I guess what makes it unique there is that there is actually access to these other immune cells like macrophages, T cells. I guess in the central nervous system, the main or only immune cell is microglia. So could the neuroimmune interactions be a bit more complex peripherally?
No, not necessarily. There are several other players in the spinal cord and in the brain in terms of other immune cell types. It's not just microglia. So it's not that it's more complex than the periphery. It's just that the periphery is in many ways easier to study, especially at least in terms of access to human tissues. They're much easier to obtain, and we can actually perform live recordings from uh, human DRG neurons obtained from deceased donors. That is not very feasible in terms of studying the human spinal cord, for example. So I think it's just a methodological issue, mainly that we haven't developed the tools to look hard enough functionally in the spinal cord, at least in humans. And so I think as our tools get better, we'll uncover more clear mechanisms of the role of immune cells in the spinal cord, especially in the human spinal cord, as well as the ability to store and preserve human tissues for live recordings improves. That has been a game changer for the field. As that improves, we'll see greater developments in that space, I'm sure.
Okay, I just wanted to say I made a very serious error of only thinking about the brain when I said central nervous system because I study the brain. So I wanted to firstly apologize.
Oh yeah, that's fine. No, a lot of people forget about the spinal cord, even though, I
Yeah.
think of it as the first brain because I studied the spinal cord for most of my PhD, and then I moved to studying the brain for my postdoc. And we've done some things in the brain, but we think of it as once you have chronic pain and it's made its way up to the brain, it's almost too late in some ways. So understanding what mechanisms are driving peripherally chronic pain states, is what we've chosen to focus on mainly.
But I guess I learned from your paper, as far as I understand, there are a lot of immune cells surrounding the dorsal root ganglion, so in the spinal cord. But I thought that in the brain, part of the central nervous system, it's just microglia. Is that not the case?
There are other immune cell types, as far as I'm aware. I know that the blood-brain barrier becomes compromised in different disease indications, and so you can actually have other immune cell types getting in. But the meninges, for example, is kind of understudied, and there are a lot of people looking at more carefully what's going on. And microglia, they seem to be involved in everything from Alzheimer's to chronic pain, different states. But there are clearly other things going on, and we've seen some somewhat promising therapeutics targeting microglia for Alzheimer's fail miserably, because they are important in things like maintenance of blood vessels and things like that. So you can't just target them as this sort of pathological thing. So it's a very difficult balancing act. And I think, that's why the periphery is still extremely attractive for drug development purposes because you're outside of the central nervous system and you're less likely to have this horrible side effect profile. So having things that are peripherally restricted that can stop chronic pain are still very attractive to the field.
Yeah, that's a lot clearer. Thank you.
For someone that's not in the field of pain or immunology, could you explain a little bit what is the relationship between pain and the immune system? Is it that the immune system helps subside the pain? Is it that the immune system exacerbates it? How is it involved?
Yeah, so this is going back to some old literature. So, the general sort of phenotype of pain and chronic pain is that people will experience an overall malaise. They'll almost have a sickly phenotype, and sort of that feeds into the sort of what might be driving that, and that had kind of started the whole thought process behind, okay, perhaps the immune system is involved in chronic pain states, that people are, running a fever and they're almost like they have some kind of infection. So perhaps they might be attributed to the role of immune cells. And so there's a lot of different angles to this problem. It's not just that chronic pain, resulting from an injury is going to involve different immune cell types recruited, releasing cytokines, sensitizing the afferents and driving chronic pain states. There's a number of studies more recently on the role of autoantibodies. People with autoimmune disorders, for example, will experience chronic pain. So those autoantibodies obviously are released by immune cells, and they're going to bind to different receptors directly on the neurons and actually sensitize the neurons directly. And so as a non-immunologist, I won't go into too much detail about what specific receptors and so forth are involved in specific pain pathways. Suffice it to say, they're important, and it's an active area of research. And I think it started with this sort of idea that, people with chronic pain have the appearance of being someone who has an infection. And so that kind of started the whole idea that this probably is driven partly by a crosstalk between the immune system and the nervous system.
Just to wrap up the neuroimmune side of things, given that there's a much greater percentage of women that have autoimmune disorders in comparison to men, is that something you also see reflected with chronic pain? Does it primarily affect women?
Yeah, it's higher incidence of chronic pain definitely in women than in men. And this is part of the problem with the field has for many years only studied male mice. And so
Yeah.
that's kind of why all of our studies now use equal groups of male and female mice so we can sort of get an idea of whether a therapeutic is likely to work. Another issue has been that, people will often include another group, a smaller group of female mice to do the study in, and we'll do limited studies in the female mice. And if the superficial result is similar, they'll no longer proceed with the female mice studies. But the issue is that you have to have these studies running in parallel and do the same level of rigor in both male and female mice because, as with anything, the devil is in the details. Especially if you're looking at things like mechanical hypersensitivity, you look at Paul withdrawal threshold and you have a readout that's extremely binary, but you miss perhaps other modalities that are involved in chronic pain. And so, the next generation of pain researchers is definitely doing a better job at trying to establish these differences.
Yeah,
And
that's
that's,
really good to hear. We recently did a podcast episode on autoimmune disorders, and yeah, we found out how one of the key mechanisms you could only like see in female mice, and so the previous, few decades of research with male mice, no wonder they were hitting a wall.
yeah, makes sense.
I was wondering, well, again, I know this isn't exactly your research, but being in the field, I was wondering, what do you advise for an individual that is handling chronic pain? Do you have any kind of advice or tips?
Yeah, so I'm not a clinician, so don't take this as sort of medical advice, but I will say, as somebody with chronic pain and somebody who studies chronic pain, is that keeping active is incredibly important, depending, of course, on what type of chronic pain. But keeping active is a very important part of staving off chronic pain. And so there are many studies that have linked neurotrophic factors that are upregulated during exercise to repair mechanisms and overall just mood can drastically affect chronic pain pathophysiology. Of course, it's associated with higher rates of depression and anxiety. And so if you are treating some of the key comorbidities, like depression and anxiety with things like exercise and movement, it can generally help with that sort of thing. And of course, non-opioid therapeutics, for the most part, we're actually looking more closely at the mechanisms of a natural product for treating chronic pain to try and understand it from a more modern angle, something that's used to treat chronic pain over the counter and has been for many years in alternative medicine, and try and understand how those things work. Some people swear by different remedies, and so we want to try and understand why these things might work, or at least are reported to work, and use modern approaches to help understand those mechanisms. Yeah, that's about it.
The last thing I wanted to ask is just addressing treatment. So actually linked to what you just said. But again, with the focus of the conversation a lot on the electrophysiological properties of these neurons, is your research program more focused on trying to come up with new therapies based, basically, should we be targeting the electrophysiological properties of these neurons? Or are you more interested in like understanding how existing treatments, even maybe more alternative approaches, or what we see as more holistic treatments, how they might modulate pain on the mechanical cellular level?
Yeah, it's sort of both. So the problem is that standard of care treatments sometimes work, sometimes don't. And so now with the rigor of more sophisticated approaches like electrophysiology and transcriptomics, things like sequencing, we're able to bridge that gap and sort of figure out why things might work and why things don't work. And so based on that, we can uncover new targets and then validate those targets and then go back and start the pipeline again for development of novel therapeutics. So we do definitely inform our research program based on both approved therapeutics as well as what's going on in the actual model and how to alleviate pain.
That makes sense.
I think it's really cool that you are not only researching current ways to help treat pain, but also like modern and over the counters and perhaps like natural remedies. Because, yes, I think there's so much colloquial information, but I also think it's important to actually research those things and see if there's like some truth in it. I'm curious about what is the state of funding for topics like chronic pain. Because, for example, for many issues that primarily affect women, there tends to be less funding, like in autoimmune disorders and things like that. What is it like for chronic pain?
I've been very lucky to work in the US during a time when, they have invested a lot of money in pain research, especially. The NIH HEAL initiative has been pretty instrumental in funding a lot of my research and the Department of Defense as well. So I think there's been sort of a paradigm shift in the funding for chronic pain and trying to understand sex-specific mechanisms. And even during my time in the UK, there was funding from the Wellcome Trust I had through my mentor to help understand chronic pain mechanisms. So I think funding agencies definitely get it that we need to invest more in understanding sex-specific mechanisms for understanding chronic pain. But I will say that because of the opioid pandemic in the US, especially, that's been especially hard hit, there has been a lot of investment in pain research.
That's good to hear.
Awesome. I guess we'll wrap up the episode there. Thank you so much for joining us and answering all of our many questions on pain.
Great. All right. Thanks for having me.
Thank you so much for joining us today for another Tools for Looking into the Brain episode. And thank you to Scientifica for supporting this series. If you're looking for more information about electrophysiology or support to set up electrophysiological systems in your lab, do check out Scientifica's website, scientifica.uk.com. And if you're interested in hearing more about the variety of research methods that are used to understand all aspects of neurobiology, check out the rest of our series. You can find more information on our website, neuroversepod.com, and follow us on social media at neuroverse_pod for updates.